Thank you to Aspire for inviting me to give this lecture on sperm selection techniques. My name is George Liperis. I am the Deputy Scientific Director at Westmead Fertility Center and a clinical senior lecturer at the University of Sydney. So what we're going to cover on this lecture is, first, I'm going to give a quick overview of the male infertility, and then move on to describe the sperm preparation and selection techniques. And after that, we're going to evaluate the effectiveness and safety of those advanced sperm selection techniques for use in the clinic. So just a quick overview of the male infertility. We know that 1 in 6 couples worldwide experiences some form of infertility. And for women, that ranges between 8 and 12% worldwide for women aged 20 to 44. And 20 to 30% of those infertility cases are explained by physiological causes in men, 20 to 35% by physiological causes in women. And there is about 25 to 40% where there is a mixture of problems between both partners. This is based on the ESHRE heart fact sheet. So there is a relatively low efficiency of ART, and that could be partly due to the quality of the spermatozoa, including poor quality ejaculates, as well as testicular samples, as well as high percentages of DNA fragmented spermatozoa and ejaculates. So the spermatozoa itself and the origin of those is quite important. So male infertility is the most common cause of infertility, and we know that up to 50% combined of couples will have a male component in the problem. And there's also lately increased cases referred due to improved techniques for treatment, such as the open microdissection techniques. And causes of male infertility can vary, and we all know about the poor semen quality, the ejaculatory dysfunction issues, obstructive as well as non-obstructive azoospermia reasons. And of course, in the ART setting, we rely on the WHO criteria to assess semen quality. So semen quality is a measure of male fecundity, and there are reference intervals for values of semen parameters that have been derived from the fertile population, and that provides the data from which prognosis of fertility, diagnosis of infertility can be extrapolated. So we can see here the WHO criteria along with their lower reference limits for the semen characteristics in sperm. And as we can see, there's a few different characteristics, and most of us are familiar with those, like the volume, the total number of sperm, the concentration, total motility, progressive motility, vitality, as well as sperm morphology. But we can see that there has been evolution of a normal semen parameters values of the different WHO manuals from the 1980 to the 5th edition in 2010. And if we can just compare between the previous edition and this edition, we can see that the majority of the reference values have been reduced, from the volume to the sperm concentration, total sperm count, motility, and morphology have all been reduced. This is because the sperm counts are declining. There have been a few studies that have indicated a worldwide decline of sperm counts, including this study in the US, a global study, European study, and French sperm quality. However, we have to understand that in these earlier studies, the techniques were less developed and the results were less reliable. And the sperm levels can drop a lot away, and that doesn't necessarily mean that it will affect the men's ability to fertilize an oocyte. Also, the duration of abstinence plays an important role, and as we can see in this diagram, we can see that the duration of abstinence has a positive effect on the testicular volume and therefore the total sperm count. So when taking this into context, retrospective or short-lived studies cannot provide substantial evidence to support declining sperm counts. And only when we have a prospective monitoring system, we can make such an assumption. So we can see here in this study that we can see the mean sperm concentration of 5,000 Danish army draftees over a span of 14 years. We can see that there is a little bit of variance from year to year, but we cannot really extrapolate that there is a decline in the sperm counts. Also, in a different study of analyzing semen parameters in young healthy volunteers every month, Based on those results, they have suggested that multiple and considerable semen analyses are needed when evaluating semen parameters. So therefore, the sperm is not declining. So we therefore need to identify and try to find ways of preparing and selecting which are the good sperm. And in the clinical setting and in the everyday IVF practice, there is two major techniques for the processing of the ejaculated sperm. Those are by means of gradient centrifugation and by swim up. So these preparations, both preparations outcome is to enrich the motile sperm population via reducing the majority of non-progressive sperm, as well as immotile and non-sperm cells. So this enriched population is then suspended in capacitation medium and incubated for several hours prior to downstream applications like conventional IVF or ICSI. So here we can see how sperm preparation by the discontinuous density gradient centrifugation is being done. Usually the discontinuous means two phases, a lower phase that usually has between 80 and 90% concentration and an upper phase between 40 and 50%. We place the semen sample on the top, and here we can see that after the initial centrifugation for 20 minutes, we can see that the seminal plasma remains above. We have the white blood cells being trapped before the upper phase. All the abnormal and non-motile sperm, or should I say the majority of those cells, are being situated in the upper phase. And following the interphase in the lower phase, we should have a high percentage of viable motile sperm. So following that, we aspirate the pellet and resuspend it in wash media, followed by another cycle of centrifugation, followed by another aspiration of the pellet and a further resuspension and another wash. And after the wash, the pellet is finally aspirated and resuspended in the IVF media for the downstream applications. So from the final stock, the concentration of the sperm is being assessed, as well as the motility, and usually samples, and that's our practice. Of course, every clinic has different practices. We consider that samples with more than 1 million per ml of progressively motile sperm, which is more than 70% of the sperm population, can be used for conventional IVF, and the rest of the samples can be used for ICSI. Of course, the swim-up method is a process of self-selection based on the ability of sperm to swim. It does also involve centrifugation, where the culture medium is first mixed with the liquefied sperm and the supernatant removed after centrifugation, and then the culture medium is smoothly layered over the cell pellet, and the motile spermatozoa are allowed to swim up into the medium during incubation period. The upper part of the layer medium is carefully separated and used for downstream applications once again. So which of these two studies should we use more to prepare? In this study, we can see that it was published a couple of years back, that we can see that in terms of fertilization and utilization, with either the sample being used for IVF or ICSI application, and this includes normal semen, abnormal semen, as well as oligoastheneteratozoospermic samples, there were not major differences between the two techniques. So therefore, selection and preparation does not depend on either we do gradient or swim up. Another thing that we have to take into account is that all sperm samples have a complex heterogeneity. And both methodologies for sperm preparation are based on the ability of sperm to swim, and that does not translate into higher quality sperm. It just means that higher yield of motile sperm. So it has been pointed that both of these techniques, swim up and density gradient, are not efficient methods to select spermatozoa in terms of apoptosis, DNA integrity, membrane maturation, and sperm ultrastructure. So also the centrifugation steps that both of these techniques have in common can generate reactive oxygen species that can have a negative effect on sperm quality. So therefore, we should have newly based techniques that take into account the sperm characteristics and those correlated to the ability of sperm to fertilize, so test functionality of the sperm and the quality of the spermatozoa to support further downstream embryonic development. One of these techniques is the magnetic activated cell sorting, or otherwise known as MAX. So this method is a method that allows the separation of the cell population based on surface antigens. So the magnetic nanoparticles that are being used are coated with annexing 5, and this molecule shows a high affinity for phosphatidylserine bind to apoptotic spermatozoa. So apoptotic spermatozoa will have this substance, and those can be retained in the MAX column, therefore allowing the recovery of non-apoptotic spermatozoa for the use in ART. As we can see here, with red are the apoptotic spermatozoa that coated with annexin 5, and the semen passes through, and the non-apoptotic spermatozoa will swim through the column. The second method that takes into account the sperm membrane is the hyaluronic acid binding. And this is one of the main components of the extracellular matrix that surrounds the cumulus oocyte complex. And spermatozoa that undergo spermatogenesis and maturation all have binding sites to hyaluronic acid. And therefore, there are two commercially available methodologies of sperm selection based on the interaction between spermatozoa and hyaluronic acid. And those are the use of hyaluronic acid coated dishes, as we can see in this image over here, or the ability to pick up spermatozoa moving slower when moving into a solution containing hyaluronic acid. So here we can see this method of the dish and containing hyaluronic acid with spermatozoa binding on the surface of the dish, and therefore those spermatozoa being used for downstream application like ICSI, after showing that they have those binding ability to hyaluronic acid. The third method that is based on the sperm membrane is called the Zeta method. And this is based on the negatively charged sperm membrane. And again, there is two methods. One is collection of charged spermatozoa that adhere to the wall of a centrifuge tube. Or the second method, which is collection of charged spermatozoa migrating within an electric field, as we can see in this image on the right. So both methodologies have been shown to retain sperm with lower DNA fragmentation compared to hyaluronic acid methods, as well as when there has been a combination of density gradient on its own or max with density gradient. So as we can see here, the sperm is placed into a well that is isolated from the next well by this porous membrane that we can see in the middle. And the empty membrane is filled with medium. Then we apply the electric field with the anode in the well without the cells, and the cathode in the well with the sperms that we can see here. And due to the negative charge of the sperm membrane, the spermatozoa migrate towards the empty compartment through the porous membrane. So these are the techniques that are based on the membrane of the sperm. However, as embryologists, most of us are familiar on the sperm selection based on morphology, so under the ICSI when we select the sperm. And this is based on our ability to find good morphology sperm in terms of the head, the midpiece, and the tail, as well as the acrosome being around 40 to 70% of the size of the head. Of course, when we have to select the sperm, it all depends on what kind of sample we're looking at. We can only select the best sperm that is available on each sample. So therefore, that has to be taken into account. And another technique based on morphology has been introduced, which is called intracytoplasmic morphologically selected sperm injection, or IMSI, which is done under a higher magnification in comparison with ICSI. So we can see here the ICSI magnification, a common ICSI magnification that under which ICSI is being done, and the ICSI magnification, which is a lot more, is around 6,000 time magnification. So this technique is based on the macroscopic evaluation of cell organisms of the sperm, or EMSOM. And in a recent study, it was which was 873 sperm samples. The presence of the head vacuoles in the sperm, as we can see here, is a couple of head vacuoles, was not associated to the DNA fragmentation and live birth rates. And vacuoles were presented to be physiological features, not affecting functionality of the sperm. Of course, more randomized control studies are needed, and we're going to cover that into the effectiveness of IMSI. Also, when we evaluate sperm, the sperm viability is an important parameter that we consider because it is important for fertilization and embryo development. And sometimes unejaculated sperm, for example, on Carragher cases, or testicular sperm can sometimes result in only immotile sperm being retrieved. So immotile sperm are not necessarily non-viable. So it is important to identify which sperm are viable amongst the immotile sperm population. And there are a few ways to undertake this test for viability. So one method is the sperm tail flexibility test, which we know that dead sperm have stiff sperm tails. So therefore, this test is aimed as a good way to apply to testicular sperm preparations. We have a testicular preparation in a droplet of handling media in the ICSI dish, and the sperm which can bend and have flexible tails are identified by touching the ICSI needle to establish whether the sperm is moving or if the tail can bend. And therefore, those sperm can be used for ICSI. We also have the hyposmotic swelling test, in which the host test solution is being made by 50% handling medium and 50% deionized water. And this is mostly used for ejaculated sperm. We have a drop of host medium placed into the ICSI dish along with a sperm drop, a wash drop, and a small drop for sperm collection. And the single sperm is being aspirated through the ICSI needle, and then the tail released into the host drop. So if the sperm tail curls, that demonstrates the positive host reaction, and therefore that sperm can be used for downstream applications. The laser test can also be used in a similar manner. The laser focus placed near the sperm tail, and using a low pulse length, applying a single shot. If sperm bends or curls its tail, that means that sperm is viable and can be used again for the ICSI. And lastly, the chemical tests, like the use of pentoxifylline or theophylline, can be used to increase motility. These agents have a limited time that they can last, so it's important to identify the moving sperm quite quickly, usually with a 10% volume of this chemical agent added into the sperm drop with handling medium. And then identifying after 5-10 minutes which sperm are motile and using those for the injection. Of course, other than the morphology mechanisms, there is also sperm selection by the guidance mechanisms. So these are more physiological, and they try to resemble the guidance within the female genital tract. And that could be a more physiological way of selecting spermatozoa with fertilizing capacity and better embryonic development. So there have been 3 methods proposed. Those are rheotaxis, chemotaxis, and thermotaxis. However, there is very limited publications on using these mechanisms to improve clinical outcomes in ART. However, we want to describe those mechanisms. So first we can see rheotaxis. We can see that the flow by blue arrows is created through a series of microchannels towards a well where the sperm sample is placed. So in response to the sperm, to the flow, the sperm will swim towards it, passing through the microchannel. So this again resembles how the sperm enters and moves from the vagina into the uterus. In the chemotaxis methods, we have 2 wells connected by 2 millimeter length but 2 millimeter diameter tube. One well is filled with the chemoattractant molecule, as we can see here in the blue color, and the other well is filled with the sperm. The chemoattractant diffuses through the connecting tube, and the sperm migrates towards the higher concentration. So once again, this uses the physiology of the sperm when it approaches the COC, and that is again through chemoattractant molecule. Finally, we have the thermotaxis, in which sperm are placed on a drop of medium connected by capillary to a second drop free of cells. and we apply a temperature gradient, and the sperm responds and migrates toward the warmer temperature, which once again indicates how the sperm will move in the fallopian tube towards the ampulla. So these microfluidic systems have been tested for sperm selection using these methods. A novel study has reported selection of human spermatozoa with low DNA fragmentation by using one type of these diffuser type microfluidic sperm sorter. And there are commercially available devices in the market that have demonstrated selection of spermatozoa with low DNA fragmentation. However, only one study has applied microfluidics so far in a clinical setting for selection of sperm in an ART setting, in which again a commercially available device was used, and that showed higher quality embryos being developed, but there was no differences in fertilization, pregnancy, and live birth rates compared with SWIMAB. So they had 61 patients in each arm, all with unknown etiology infertility. So are any of these techniques effective, and have they been proven to be safe when used in the clinical setting? So advanced sperm selection techniques are meant to improve the chance that structurally intact and mature sperm with high DNA integrity are selected for fertilization. So therefore the aim is to select the sperm that can improve the genetic integrity of the sperm, the fertilization capacity, embryo production, in vitro survival, clinical pregnancy rates, delivery rates, and also demonstrate safety. So how about advanced sperm selection techniques as we have described them? There was a recent release in the Cochrane library last year in which they had 8 randomized controlled trials and a total of 4,147 women. And in this Cochrane study, there was 4 studies that compared hyaluronic acid against ICSI. One study compared hyaluronic acid, ICSI versus sperm slow, so the 2 methods using hyaluronic acid. One study compared hyaluronic acid binding versus max versus ICSI. 3 studies just compared max with ICSI, and 1 study compared selection by zeta versus ICSI. 6 of these studies reported on live birth, 7 reported clinical pregnancy, 6 reported miscarriage per clinical pregnancy and per woman randomly assigned. However, none reported on fetal abnormalities, which is again the safety element that we need to take into account. So here we can see the summary of this Cochrane study. So here we can see the comparison between hyaluronic acid binding compared to ICSI. And we can see that there was no difference in terms of the results, in terms of clinical pregnancy per woman randomly assigned. However, one important element as well is that the quality of the evidence was of low quality. And that was because of different reasons: imprecision, low event rate, risk of bias, of course, where performance bias, and therefore further trials being recommended. The next arm of the study compared hyaluronic acid ICSI compared to sperm slow, and once again they found no difference in terms of the clinical outcomes, and again the quality of the evidence was very low, again due to serious imprecision data that were derived, single randomized controlled trial, no numbers, and broad confidence interval. So therefore that study cannot provide significant evidence of the benefit of that hyaluronic acid. So when compared max sorting against the ICSI, once again, there were no difference in the clinical outcomes, and the quality of the evidence was also very low. Again, low numbers, only 2 randomized controlled trials, imprecisions, and unexplained heterogeneity between the participants. This is an interesting outcome. When they compare the zeta sperm selection against ICSI, There was a slight increase of the clinical pregnancies with using the zeta method, but once again, we have to be very cautious when interpreting these results, as the data are derived from a single randomized control trial. And once again, were very serious imprecisions, low numbers, and broad CI. When they compared the MAX against the hyaluronic acid, there were no differences. And once again, the quality of the evidence was very low. And when comparing, lastly, in the other Cochrane library review that was published this year, comparing IMSI with ICSI, they used 13 randomized controlled trials that had a total of 2,775 couples. The current evidence from the randomized controlled trials did not support or refute the clinical use of intracytoplasmic sperm injection using IMSI. And there was uncertainties towards the chances of having a live birth and the risk of having a miscarriage using this method. Once again, there was very low quality evidence that IMSI may increase the chances of a clinical pregnancy, which means that uncertainty about any real difference can be made. And there were no trials reporting the risk of congenital abnormalities. So once again, no safety data with this comparison. So therefore, well-designed and sufficiently powered trials are still required for these comparisons to be made. And here we can see the results. Once again, no real significant differences in the clinical outcomes. Once again, very low quality of evidence. So the key results from these reviews suggest that advanced sperm selection strategies may not result in an increase in the likelihood of live birth, at least as we see it thus far. The only sperm selection technique that potentially increases live birth and clinical pregnancy rate is zeta sperm selection, yet these results are from a single study and therefore have to be taken into account, and caution is required before being applied into regular and routine clinical practice. There is low quality evidence that hyaluronic acid ICSI decreases miscarriage rates when compared with ICSI, and it is uncertain whether other sperm selection techniques alter clinical pregnancy. The main limitations of these studies were imprecisions associated with low number of participants or events and high risk of performance bias. Data on important clinical outcomes such as fetal abnormalities were absent. So further studies are required before recommendations for use of these techniques are made for clinical practice. So in conclusion, sperm selection is an important factor for improving clinical outcome. The embryologist is the key, and the training is crucial when it comes to morphological selection of sperm, and the variance of experience is a limiting factor as well in comparative studies. The methodologies developed thus far have not been proven to be effective for routine application in clinical practice, but they can be promising for specific cases of male infertility. The new microfluidic systems in place have promising results, but will need to be confirmed in large randomized control trials. And it is possible that in the future, there will be multiple sperm selection methodologies applied in order to improve clinical outcome. Thank you very much for listening.